In this lecture, I'm going to talk about immune responses to viral infection. And first, I will speak brief on innate immune responses to viral infection. And as you know that the innate immune system or innate immunity is a basic immunity we born with and actually does not require antibodies. So the innate immune responses to viral infection, it is mainly through induction of type 1 interferons and activation of natural killer cell. So if human cells get infected by viruses, so the infected cells will produce type 1 interferons to protect uninfected cells, and these are induced during double-stranded RNA production, and this can induce expression of type 1 interferons by the infected cell. This is actually virally infected cells, so they produce type 1. They have receptors on other cells, for example, natural killer cells. So also the specific immune system, namely antibodies, they play also a role in protection against viruses. But however, in some viral infection, maybe antibodies produce at day 50 or at day 100, and so the immunity to viruses usually start as cell-mediated immunity, but the cell-mediated immunity has no capacity to clear and to clean up all the viruses. So appearance of antibodies is very crucial for cleaning up and removal of the viruses from the body. So these antibodies, they may neutralize or interfere with binding of viruses to the following molecules on the surface. And as you know that antibodies work by different ways, mainly 3 ways. It is neutralization, opsonization, or activation of the complement. And so here, for example, influenza virus, it binds to sialic acid residues in cell membrane, glycoproteins, and glycolipids. So antibodies interfere with this binding, and also in case of rhinovirus, so antibodies interferes between the virus and their binding site, which is ICAMs or intercellular adhesion molecules. Epstein-Barr virus, it binds to type 2 complement receptors on B cell, and so the neutralizing antibodies, it interferes with such binding. So however, in case of gastrointestinal tract, of course, very important antibody is secreted in the gastrointestinal tract, which is Secretory IgA. It is in all mucus secretion and play very important role in protection of the mucous membrane all over the body. So these antibodies actually interfere with virus attachment to the mucosal epithelial cells. So the advantage, and from here we know that the advantages or the advantage of attenuated oral polio vaccine is that it induces protection of secretory IgA, which effectively blocks attachment of polio virus along the gastrointestinal tract. And as can be seen here, this is IgA antibodies. It is actually dimeric with a secretory molecular chain, and this secretory for IgA, and actually this is a mucosa and submucosa or laminar propria. These antibodies are produced and they reach the lumen to prevent all kinds of infection involving viral infection. So as can be seen here, these are antibodies and these are virus actually. So these antibodies or the antibodies, they have the capacity to block attachment of this virus to some epitopes that necessary to mediate fusion of the viral envelope. Of course, it starts the infection by fusion, by attachment, and then by fusion of the envelope to the plasma membrane. So, but however, some antibodies also, as I mentioned, that antibodies work by 3 ways: complement activation, one of them, opsonization and neutralization. So some antibodies have the capacity to activate the classical pathway of the complement and induce lysis of enveloped virions. So these are antibodies in green. So when they capture these virions, so this will lead to formation of immune complex, and immune complex activate the classical pathway by activating complement component number 1, and this is now C1q. It binds to the immune complex, and binding of C1q with the immune complex activate C4 and C2, and then these their big fragments, or the big fragment C4b and C2b will act as an enzyme known as C3 convertase, and this will act on C3 and cleave it into C3a, C3b. So C3b will bind, and the whole molecule will act as C5 convertase, and this will lead to cleave of C5. So following that, following the binding of C5b, C6 will attach, will join, and C7 will bind, C8 will bind C9. So this will lead to formation of what is so-called membrane attack complex, or MAC, and also it will lead to formation of anaphylotoxins. This will mediate an inflammatory environment or milieu, which makes the area unfavorable for the viruses to stay and to replicate. And this MAC has also the capacity to lysis of the enveloped virions. And as can be seen here, also one of the most effective method to eliminate virus is that this is now the virus. For example, it is captured by these antibodies, and then these antibodies opsonize this virus to phagocytic cell. And as you know that the phagocytic cells, they express some receptors specific for IgE antibodies. For example, this is Fc gamma R1. It is specific for IgG1, and Fc gamma R3 specific for IgG3. And so these ones they bind, this will help in phagocytosis of this virus. So this is antibody dependent phagocytosis. But however, in some instances, the complement components C3b, they bind to the virus, and this virus now is able to bind to specific receptors, for example for C3b on the surface of the phagocytic cell, and overcome the negativity on the surface of the phagocytic cell. So now the phagocytic cell is capable of phagocytosing the virus through the C3b receptor, or it is C3b mediated phagocytosis. This is clear here actually. These are complementary receptor, and here's the virus opsonized with C3b to the monocyte. So now the monocyte can phagocytose the virus through this way. So now I am going to talk about cell-mediated immunity to virus. As I mentioned, that it is actually two cells that interfere following viral infection. It is natural killer cells, or NK cell, and CD8 T cytotoxic cells. But however, CD4 or T helper cells, they also play a very crucial role in immunity to viruses. And as you know that T helper 1, they actually coordinate or interact with the antigen presenting cell, and this interaction result in expression of very important cytokines. For example, interleukin number 2. This interleukin number 2 actually it has an autocrine function. It activates this T cell, and also it has a capacity to activate the adjacent other cells, for example CD8 T cells. So this we know it is known as paracrine cell, paracrine function. And also CD8 may early actually produce cytokines. For example, they produce interferon gamma, and they produce tumor necrosis factor alpha, which they have antiviral activity. And also the CD8 or the CD4 cell, as can be seen here, they produce interferon gamma, and this interferon, it has a viral also activity, but this is however type 2 interferons. And as I mentioned, that type 1 it is interferon alpha, beta, and maybe omega. So now let us go to cell-mediated also immunity, so activated as I mentioned that. But however, IL-2 signals are from T helper 1 also they play a very crucial role in formation of long-lived CD8 cell memory. So they are very important for this process. So it is not only CD8 and NK cell, but also T helper cell play a very crucial role in immunity against viruses. So the activated CD8 T cell also they play a crucial role in defense against viruses, and this has been demonstrated by transfer or adoptive transfer of cytotoxic T cell to non-immune recipient. So their protective role in protecting against a virus has been shown by this transfer experiment. So, and let us now come to the natural killer cell. As I mentioned that IL-2 and interferon activate natural killer cell. But however, natural killer cell also it has capacity to produce some other materials, for example perforin and granzymes. So they play actually very important role in the host defense during the first days of many viral infection until a specific CD8 T cytotoxic response develop. And as can be seen here, this is viral infected cell, but however, still the viral proteins are expressed in association with a major histocompatibility class 1. So no release of perforin or enzyme, and most probably the natural killer cell will produce some cytokines that they have antiviral activity. But however, usually viruses, they follow some strategies to evade or to escape immune responses. For example, downregulating expression of major histocompatibility class 1. And as can be seen here, this absence of major histocompatibility class 1 will be sensed by inhibitory receptors. So the natural killer cell will get activated and they will produce perforin and granzyme and will kill the virally infected cell, which is known as target cell, by a process known as apoptosis. So now I'm going to talk about how viruses escape from effective immune responses. We call it immunoevasion. So I will show just some examples. For example, hepatitis C virus overcomes the antiviral effect of interferons by blocking protein kinase R. And this is very important for the, as we mentioned, that it is type 1 interferon, the immunity against the viruses. So it has blocking and interference activity with this important protein. And also herpes virus type 1 and 2 express also proteins that inhibit antigen processing and also inhibit presentation. And as can be seen here, this is now viral infected cell, for example, so the antigens are presented in association with major histocompatibility class 1. So now it will be recognized that this cell is infected by a virus. So some viruses, in order to abolish this or to hide themselves and not to be seen by other immune cells, they tend to inhibit antigen presentation, processing and antigen presentation of viral antigen to CD8 T cells. And adenovirus and cytomegalovirus, for example, reduce expression of class 1 molecules, and actually also this inhibit antigen presentation to CD8 T cells. Here, this is one, for example, and this is CD8 T cell. This is major histocompatibility. So any antigen presented with major histocompatibility class 1 will be seen by CD8 T cells, and it will get actually activated. So also cytomegalovirus, measles virus, and HIV reduce level of class 2 and not class 1. And actually class 2 molecules are very important, as I mentioned in the beginning, that class 2, they play very crucial role as T helper 1 usually recognize antigens in association with major histocompatibility class 2, and so it will produce interleukin number 2, and which is very important, and it has this cytokine a paracrine effect on CD8, and it makes it of long life, and it makes it get activated, and it will show its antiviral activity. So cytomegalovirus, they know this information, and also measles virus and HIV. So they reduce level of these uh, uh, molecules of class 2 on the surface of the antigen presenting cells. So now I will show how some viruses evade the effective immune responses, or what is so-called immune evasion, by inhibition of complement components. So, for example, vaccinia virus and herpes simplex virus, and these, they bind to C3b. This is here C3b, C3b and C4b, and this interfere with the complement pathway, interfere with formation of membrane attack complex, interfere with formation of anaphylatoxins, which are very important for lysis and important for the inflammation during infection. And however, some viruses may produce immunosuppression, and it will lead to suppression or cause generalized immunosuppression. For example, Epstein-Barr virus or cytomegalovirus and HIV. And in case of actually Epstein-Barr virus, they produce cytokine known as IL-10-like. It is not an IL-10, but it is very similar and homologous to IL-10. And as you know, that IL-10 has the capacity to inhibit macrophage, and macrophage is very important because they produce pro-inflammatory cytokines like human necrosis factor alpha, interleukin number 1, and also it produces interleukin number 12, which is very important for activation of T helper 1. And activated, as you know, activated T helper 1 will produce in interferon gamma, which has anti-viral effect. So the Epstein-Barr virus, they produce this material. It is IL-10-like, and this will suppress the expression or secretion of cytokines by other cells. So also one of the most famous and effective ways for the virus to evade or to escape effective immune responses, known as antigenic drift and antigenic shift. And as you know that if we go deep in the virus and to speak about the virus genome, the virus actually, the genome, it is formed of blocks. So these gene blocks, the virus, when it is under attack, may give an order to gene blocks to change their order. So gene number 1 will sit in the place of gene number 7, and block number 7 will come and sit on the block number 1 within the gene locus, and this will produce a new antigen. It is known as antigenic drift. So the immune system will start the defense from the beginning, and when these viruses get recognized by the immune system, so also the virus will give order for an extra process of antigenic drift. But however, this may take 5 years of time. They deal with the immune system and with the environment, and after 5 to 10 years, maybe 12 years in case of corona, so the virus give an order for the genes that make possible target for killing by the immune system, give an order just to get down regulated and to upregulate very new gene, and this what may lead to outbreak of a disease like what happened in case of corona virus. So also influenza virus, it is follows this way, the antigenic drift and antigenic shift. So Antigenic drift, it is actually a matter of deletion, as I mentioned, or insertion or change in the blocks of the gene within the locus. So this actually has led to emerge of new subtypes of, for example, viruses or influenza virus, and this may be known, may occur through genetic reassortment. Assortment means exchange of, in some instances, it is not necessarily for the virus to upregulate new proteins from new genes. But in some instances, these viruses may exchange their genes. For example, in the human, may exchange their genes with a similar virus that affect animals, and this is known as genetic reassortment. Thank you very much, Professor Ahmed Bolan.